Hello everyone. Am I audible and is the screen visible?
Yes,
Okay, thank you. So we will begin in another 2 minutes. We will begin our today's session. So welcome everyone to the NPTEL course on post-harvest operations and processing of fruits, vegetables, spices, and plantation crop products. So this course is instructed by Professor H.N. Mishra, who is a professor in the Department of Agriculture and Food Engineering, IIT Kharagpur. I am Shikha Sharma and I am the teaching assistant for this course for July 2025 semester, and I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So this live interaction session is a week 3 live interaction session now, and its assignment deadline is on 13th. So uh, in this live interaction session, we will be discussing these topics, which is module 3 topics. So which includes basics of processing and preservation of horticulture and plantation crops products. And then we will discuss about processing by removal of water, by processing by the addition of heat, processing by removal of heat, irradiation of fruits, vegetables, and spices. So this is the module 3. So it is having 5 lecture videos, lecture 11 to 15. So it includes basics of processing and preservation, processing of processing by removal of water, processing by addition of heat, and processing by removal of heat, and about irradiation of fruits, vegetables, and spices. So these 5 lecture videos, I hope you have all gone through all these 5 videos. So uh, we have a tutorial class on this. So let's solve this our question on these 5 topics. So this is our first question for today's lecture, which is which of the following statements are correct about the control of pH in food? So we know that the microorganisms basically they grow in the food and they spoil the food products, right? They spoil the food products. So we want to prevent the microbial growth in the food products. So those microorganisms, their growth, they are basically affected on various parameters like intrinsic parameters, which are like the parameters which are related to food products. So factors inherent to the food like the food chemical and physical properties of the food. So which includes like pH, moisture content, oxidation reduction potential inside the food, nutritional content, natural antimicrobial present in the food, and then biological structure and natural microflora of the food. So these are the intrinsic parameters which affects the microbial the wild or the spoilage causing microorganism or the pathogens in the food. So if the pH will be uh, very low and very high, then certain kind of bacteria cannot grow. And if the moisture is very low, then the microbial growth will be very low. So further the spoilage, if the microbial growth is very low, so the spoilage will also be very low. And certain others are the extrinsic parameters, which means that these are the like the how is the environment around the food is there. So those parameters also affect the microbial spoilage in a food, like storage condition of the food, that is like property of the environment in which the food is being stored. So the outside temperature, like if the food is stored in the low temperature, or so the microbial growth will be low and the spoilage will also be very low, and spoilage will be low, right? And also the relative humidity of the environment affects. If relative humidity is very high, so the microbial growth will be very high and spoilage will easily occur. And then presence of the gases or oxygen. So if the uh, CO2 amount is more, or inert atmosphere is being provided, so all or the oxygen amount in the air, it affects the growth of the microorganisms, because we know that there are certain microorganisms which need oxygen to grow and certain cannot grow, uh, cannot grow if the oxygen is not there. So microorganisms will not grow if oxygen will not be there, right? So another kind of microorganisms, like if uh, they can, they are the one which grows in the absence of oxygen only. So they can grow when oxygen is not there. So it affects the presence of gases, or the oxygen affects the microbial growth in particular food product. Also, the antimicrobials present or the added microorganisms, they also affect the microbial spoilage of the food. So these are the certain different factors which affect the microbial spoilage in a food. So our question is about the pH, which is a intrinsic parameter in the food. So food's pH, that means, so the question says, which of the following statements is are correct about the control of pH in food? So we have four statements that pH is an important factor affecting the growth of microorganism in food because it affects microbial energy metabolism involving the buildup of gradient of H plus ions across the membrane, and microbial enzyme activity and stability of cellular macromolecules. Second is the pH can be controlled by the choice of raw materials like plant variety, animal tissues, and by the addition or in situ, or in situ means in the food original location. So, or in situ formation of acidic or alkaline low molecular weight compounds in the food if they form. So it talks about that. Third is growth of microorganisms may be inhibited by either low or high pH. Fourth statement says minimum pH for growth as well as the rate of inactivation of the microorganisms by acid is unaffected by the presence of other inhibitory factors such as low water activity, preservatives, and low temperature. So which statement is correct? Anyone? Anyone would like to answer? So Vishakha is saying A. Anyone else? Suraj is also saying A. Okay, so let's see the answer. A is correct answer. So 1, 2, and 3 are the correct options, correct statements, and fourth one is the wrong statement. So let's see that basically pH, it affects microbial energy metabolism it because what happens when there is a cell, like suppose this is a microbial cell. So if there is a very low pH and pH leak, H plus ion leak into the microbial, so this microorganisms will try to maintain the homeostasis condition and to neutralize this acid and try to maintain the pH inside the cell, inside the cell. Okay. So because of that, it needs some energy also. So its all energy will go in maintaining the balance or the homeostasis condition. So pH affects microbial energy metabolism involving the buildup of gradient of H plus ion across the membrane and microbial enzyme activity and stability of cellular macromolecules influencing the growth of microorganism in the food. So since H plus ion is coming inside the cell, so microorganism has to invest the energy to maintain the homeostasis condition or balance the pH. So that its microbial enzymes and the proteins and other cellular macromolecules will not influence. pH affects sensory properties of many foods. So if the pH is so statement one, which is this one, is correct, right? And because pH is important factor in the growth of microorganism in food, because it takes microbial energy metabolism to involving the buildup of H plus ions and microbial enzyme activity and stability for maintaining the microbial enzyme activity and stability. Uh, Microorganism will use all its energy to maintain the pH. So pH affects its growth, right? Second is that pH also like affects the sensory property in many food products. So if the pH is very low, that means highly acidic food, and we can feel the sour food product, right? pH can be controlled by choosing appropriate raw materials and by adding certain kind of acid or alkaline by low molecular weight compounds. So pH also this can be controlled by the appropriate raw materials. Either they will be added or like from outside, if can be added or can form inside of the cell, and uh, these compounds can alter the pH. So option second is also true, that we can add certain compounds from outside or can form like certain compounds will be inside only and they can in like in the citric acid, the pH is very low. Citrus fruits, the citric acid amount is very high and the pH is low. So certain kind of compounds, they will change the pH. So also, also both low and high pH can inhibit the growth of microorganisms. So microorganisms, they will grow mostly in the neutral range, 6 to 7 around. So if the and these microorganisms which grow condition, if pH will be very low or very high, they will not able to grow. But there are certain microorganisms which grow in at the low pH and at high pH also. So this statement is also correct that the pH affects growth of microorganism. But the fourth statement is not correct, as it is saying that the pH is that minimum that pH is that in hurdle technology we use certain like water activity, temperature, preservative, other factors to minimize the growth or to control the growth of the microorganism. Saying that unaffected by the presence of other inhibitory factor, which is not correct. So A is correct here. So minimum pH for the growth and the rate of inactivation by acid is indeed affected by the nature of acidulant, which acid is there, presence of others inhibitory factors like low water activity, preservatives, and low temperature. So these will also other factors, they will also complement if the pH is low, and then if other inhibitory factors will be there, they will complement, and that is the technology known as hurdle technology that may interfere with the metabolism or increase the need for maintenance of the energy and the ability of microorganism to react to acid stress to maintain passive and active pH homeostasis. So if there are the low pH and some certain other inhibitory factors like low water activity and some acidulants will be there, or the temperature is very low. So microorganism will going to need certain energy to maintain the homeostasis. So there are two kind of uh, homeostasis: passive pH homeostasis and active pH homeostasis. So they both should be maintained. So passive means that the microorganism either prevent external protons from entering into the cell, so or increasing capacity of their cytoplasm synthesizing the glutamate or citrate. So either they'll produce buffer or they will prevent the external proton to come inside the cell. So they will prevent these protons, H plus ions, to come inside. Active pH homeostasis means cell maintains their cytoplasm pH through metabolic activity. So in active pH homeostasis, cell utilizes its own material to produce the compounds which will going to react with H plus ion and neutralize. So this will be going to be a very exhaustive process for microorganism when the pH is low or pH is very high. So till the pH homeostasis is maintained inside the cell, the microorganism, they will survive. So microorganism will try always to maintain the pH either through the active or the passive pH homeostasis. So microorganism, they will try and they will fight with the stress, and so that the balance should be maintained and they can survive. So in that process, a lot of energy will go and microorganism will feel exhausted after a certain time, and their growth will be inhibited. So we can see that there are different food products like apple. So apple, it has low pH like less than 4. So microorganisms will grow less because it has malic acid in this. Tomato, its pH is also low, 4.3 to 4.9, of the presence of oxalic acid. Milk pH is generally 6.7 to 6.9. So it is not a very low acid food because of the presence of lactic acid. So like lemon basically is the most acidic food, then apple, then milk, and ammonia is basic range. From this table, we can see that there are different vegetables and what are the pH. Here are the different fruits and what is the pH of these food products. So we can see apple is a very high acid food, and we can see another food like cucumber, it is also somewhat acid. We can see that lettuce is around nearby 6. So all these products which are having near 6 pH, their microorganism will grow more compared to the less acidic food products. And we can see that this is the pH scale. So what which kind of the microorganism can grow in which pH? So mold is a very resistant one. It can grow even at the low pH. Then yeast and then different bacteria. So we can see that this is the range like from 5, 8. From 5 to 8, most of the bacteria like they and most of the microorganism they can grow easily. And in 6 to 7, it's very prominent. So is this question clear to everyone? We are moving to this next question about water activity, that which of the following microorganisms have the lowest water activity requirement for the growth. So we have 4 microorganisms which has the lowest water activity. So anyone can tell what is water activity and what is the difference between water activity and equilibrium relative humidity? There are two terms in the food very prominently used. Where microorganisms easily adapt themselves and grow 0.98 to 0.9. Yes, 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 Suraj. So water activity, if it's more than 0.97, so they can grow easily. But here the question asked low water activity. And I asked about what is water activity, difference between water activity and equilibrium relative humidity. ERHV says, right? Water activity is the measure of available water in the measure of available in food, measure of water available in the food, right? And what is equilibrium RH? Vishakha? So let's discuss what is all these terms. So water activity is basically the ratio between vapor pressure of the food itself when it is in a completely undisturbed or balanced condition, and then balance with the surrounding or air media to divide like P by P naught. So A will be P by P naught. So it will be that this P will be ratio of the P will be the vapor pressure of the food when it is in completely undisturbed or balanced with the surrounding air, and P naught will be the vapor pressure of distilled water under identical conditions. So this is in food divided by vapor pressure in food divided by vapor pressure in water. So this is water activity, while equilibrium relative humidity is the water activity into 100. So basically, equilibrium relative humidity is when like there if there is a like food product is there, so there will be some water in the food and there will be some relative humidity outside. Here it's water in the food and there will be some relative humidity in the environment. So in this particular area, which is very near to food product, so water will try to, if the amount of water will be less in uh, food, or it will, if it is very high in the food, means it is not same as per the surrounding air, so it will try to maintain the equilibrium condition, right? uh, the water will flow from high concentration to low concentration. So if water concentration is high in the food, it will flow outwards towards surrounding air. And if water is more in the surrounding air compared to food, so it will flow from air to food product, right? So there will be a certain area where the equilibrium condition will be maintained. No, so that is the equilibrium relative humidity, and the relative humidity which will be in the like outside air, which is like far than the food, it is the certain amount of the relative humidity which is just the relative humidity. Near food product it is equilibrium relative humidity, so that is the difference. So the water activity is basically increase with the temperature. Most food products, they have a water activity above 0.95, and that will provide sufficient moisture to support the growth of bacteria, yeast and mold. So we can see in this table that uh, molds can grow above 0.7 water activity, yeast from 0.88 to 0.91, and bacteria needs more water, like the highest amount of the water activity. If the water activity is greater than 0.91, they can grow. So uh, and here is the difference given, what is the moisture content in a food product and what is the water activity. So there many of the students, they got confused that uh, we says when we are doing the drying calculation and all, we use the term moisture content, and so whenever we are discussing with a microorganism, we discuss the term water activity. So moisture content is basically the bound water plus free water. So there are different, as we know that there are different kind of water in a food product. So which are the water which is very much in contact with the food is bound water, and which is little far is free water. So both the bound water plus they will add and they will be content. Whereas in water activity, we just only take the free water content in a sample. So this is the difference between water activity and, moisture content, and water activity and this relative equilibrium relative humidity. So let's see, lowest water activity we need to find, right? So lowest water activity is must be in molds, right? Certain kind of molds. So which kind of molds will have molds or yeast? Sometimes yeast also. So which kind of molds or yeast can be in low water activity range? So anyone, any guesses? A, B, C, D from which option is correct? C. Yes. So C is correct answer. Osmophilic yeasts will have, uh, they can survive in the low water activity range. So basically, osmophilic means they need, uh, like they are very, they can grow in high osmotic pressure, or they can be known as osmotolerant. So they can grow in high osmotic pressure where, like, the high amount of salt is there or high amount of sugar is there, where water activity is low, they can grow, right? So they thrive in environment with low moisture content, where P by P naught can be either 0.65 or 0.60. So they can survive even in this low water activity. Therefore, to suppress its growth, it is very crucial to lower the water activity even below 0.6. So we should have low water activity than 0.6. So as the table suggests that common spoiling bacteria, they need 0.97 water activity. Clostridia, which is the first option, it needs 0.94. Then Bacillus, which is the second option, is 0.93. And then Staphylococcus aureus, aerobiasis, and both aerobic and anaerobic. So anaerobiasis and aerobiasis, they need around 0.86 to 0.91. Yeast and molds 0.86, but the osmophilics and osmophilic yeasts and xerophilic molds, they can grow up to 0.6 water activity. So is this question, the second question is clear to everyone? Then we move to this question about blanching and other heat treatments.
In the product.
Yes. Anyone is saying anything? Any doubts? Okay, so let's move to next question because we don't have any doubt so far. So which of the following statement is correct about blanching? So blanching is used for enzyme inactivation in fruits and vegetables. Blanching is a severe thermal treatment. Blanching uses only hot air for heating the product. Blanching is typically performed at temperature below 50 degrees Celsius. So which statement is correct? Any guesses? A. Vishakha is saying A. Haribabu is saying A. Let's see answer of this question. So A is correct answer, that blanching is used for enzyme inactivation in fruits and vegetables. So blanching is basically a thermal treatment, uh, where that is usually performed prior to food processes such as drying, freezing, frying, and canning. So before all these processes, blanching can be used as the primary process. It basically it is a thermal treatment. Okay, so in this what happens, it inactivates enzymes and it destroys microorganisms that might contaminate raw vegetables and fruits during production, harvesting, and transportation. So it inactivates the enzyme, which is in statement correct. Also, it destroys microorganisms at the production, harvesting, and transport step. Whatever microorganism contaminate at that stage, when do we do the primary processing in the industry, the first as a the first step, then we do to remove some microorganisms. Okay, then it involves heating the vegetables and the fruits rapidly to a predetermined temperature. First heating will be there, the temperature high will be there, and maintaining it for certain specified amount of time, like 1 to less than 10 minutes. So 1 to 10 less than 10 minutes or 1 to 10 minutes, it will be hold this particular temperature hold, uh, and then it will be cooled down. The product will be cooled down. So it is a mild heat treatment. Blanching and pasteurization comes under mild heat treatment method. So blanching is not a severe thermal treatment, and it uses either air, water, or steam to produce heat, to heat the product. And so blanching basically use only hot air. No, it uses air, water, and steam for heating the product. And then blanching is typically performed at temperature below 50 degree. So actually blanching is performed at 88 to 99 degrees Celsius, sometimes like 70 to 99 degree also. And less than 10 minutes it will be performed. Uh, so below 50 is not correct. So option A is correct. And in blanching, we have various different technologies like hot water blanching, steam blanching, high humidity hot air impingement blanching, microwave and radio frequency blanching. So this is in the novel technology, ohmic blanching and infrared blanching. So these all are the three. One will be the novel, and these all where hot water, steam, or the uh, hot air is being taken. So they will be in conventional method. So blanching basically inactivates enzyme and enhance drying rate and product quality, expelling air inside plant tissues, reduce browning reactions. So if the air is not there, so browning will also not there, and if enzyme is deactivated, so browning will also be not there. Decreasing, and it decrease microbial load in tomatoes and such products. It is used as a peeling of the product, and it helps in removing pesticides and the toxic residues on the surface of the food product, and it increase extraction of bioactive compounds. Other purposes of blanching includes that it cleans the surface of the plant, it removes damaged seeds, foreign materials also, and it kills the parasites and its eggs on the which are on the surface of the fruits and vegetables, and it reduces sometimes oil uptake. So this kind of treatment, blanching, is used for these applications. So in certain vegetables are like given here, like pea, blanching temperature is 85 to 90 degree, and it is done for, the heat treatment is done for 2 to 7 minutes. Like carrots for 90 minutes, sorry, 90 degrees Celsius for 3 minutes. Like these, cauliflower for around like 100 degree for 2 minutes. So like these, blanching is treatment. These are the example. This table depicts example of blanching of different vegetables. But blanching has certain disadvantage also, that it loses some of the nutrients when we do hot water blanching, then the steam blanching, and sometimes the steam causes uneven blanching and results less efficiency. And blanchers, the steam blanchers, they need higher investment. So this is the disadvantage of blanching. So like nutrients, like vitamin C, which are heat sensitive nutrients, they can be lost during the process. So like this. So statement 1 is correct in the blanching. Let's move to next question. If it's clear to everyone. Blanching is clear to everyone. Is there any doubt so far? So let's move to
Hello.
pasteurization.
Hello.
Yes, yes,
Uh,
please.
yeah, ma'am. So the blanching, so which one is the correct method for blanching? The hot water is not, steam is not done.
We can go for novel technologies since these the conventional technologies were not appropriate. Sorry. So new methods they are being developed like microwave blanching or radio frequency blanching. So we can use those new technologies.
So microwave and radio frequency is there. Then ohmic blanching is what?
Yeah, so these are all the novel technologies. The microwave, ohmic, infrared. So these are the new one. Like so these technologies we can use rather going for the conventional technologies which are like hot water, steam, or using hot air like that.
Okay. So are you going to tell about this novel technologies?
In detail, you want to know? I
Yes.
have not covered in this slide in detail about these technologies, but maybe in next class I will include some slides regarding all these technologies if you want.
Okay, and one more question, ma'am. Will we get some stuff for study about it? Because now we are going through the slides and all. Will we
Hmm.
get something for the next or future reference? Like,
Uh, so like you have already the PDF of the slides which I am teaching, right? So what material you want now?
okay, so the
About
PDF is there. Actually, I don't know about that. Okay, where will I
okay.
get the PDF?
Actually, what is happening? So whatever the lectures we have covered, we covered 2 weeks, and in the Google Drive already, like in the last of this PPT, I will show you one Google Drive link. You can go there. They have like 2 week PPT already been uploaded, and this will third after the lecture. I will upload week 3 also there. The PDF all of all these PPTs that for your reference. And also, all the lectures are already recorded, and 2 week lectures are already on YouTube. So you can watch those lectures also.
Okay, and will you send something about this novel balancing methods
Yes,
slides? Will you
yes,
add?
yes, yes. So I am thinking to add in the discussion of the next week. So in week 4 PPT, we will discuss also in the class, and we will be going to include in the slides, like in the PDF which I am going to share. I will include that also.
Okay,
Is that
thank.
okay?
Yeah, thank you, ma'am.
Okay, okay. Thank you so much for asking the question. Okay, let's move to pasteurization. If no more questions, so which of the following statement Yeah, which of the following statements is correct regarding pasteurization? So pasteurization is a mild heat treatment, first statement, and second is it aims to kill all the microorganisms present in food. Third is low temperature, long time pasteurization is conducted at 63 to 65 degrees Celsius for 30 minutes. High temperature, short time pasteurization typically uses temperature of 71.7 degrees Celsius for 15 seconds. Let's see the answers. So Suraj is saying A, Vishakha is saying C, Gauri is saying C. So we have many answer. Oh, C are more. Hari Babu is saying also C. So we have many C. Let's see. B is the correct answer. Let's see why. So we know that pasteurization is a mild heat treatment. We already discussed that the blanching and pasteurization, they both are mild heat treatment. Okay, blanching and pasteurization are both mild heat treatment. So this option is correct, first one. Then that pasteurization is basically any process, treatment or combination thereof that is applied to food to reduce the most resistant microorganism. So in pasteurization, we have a aim to kill most resistant microorganism or enzyme of public health significance to a level that is not likely to present a public health risk under normal condition of distribution and storage. This definition is set by National Advisory Committee on Microbiological Criteria for Foods. So what it says is that pasteurization means the killing of most resistant microorganisms or enzymes and which are important to for the public health, and they will be killed to the certain to the count will be lowered down to certain amount which will not affect any public health. They will not be like the level is like that they will not likely to present a public health risk when product is being stored in at normal condition like room temperature or ambient condition. So but in statement second, it says that it aims to kill all microorganisms present in the food, which is not correct. So it aims to kill part of microorganisms and eliminate human pathogens, not all microorganisms. Then it includes thermal treatment like steam, hot water, uh, heating, ohmic heatings, microwave heating, infrared heating, and non-thermal also like high pressure processing, ultraviolet radiations, irradiations, pulsed electric field, chemical treatments, ultrasounds, filtration, high voltage, arc discharge, etc. So these kind of uh, methods, uh, both the thermal and non-thermal technologies, they both are included uh, in this kind of treatment, which is pasteurization. It's just not thermal one, non-thermal also, because it talks about decreasing this count of microorganism, right? So let's see this pasteurization. It is of two type: LTLT, which is low temperature, long time, and HTST, which is high temperature, short time. So LTLT is around 63 to 65 degrees Celsius for 30 minutes or 75 degrees Celsius for 8 to 10 minutes. And high HTST is at 71.7 degrees Celsius and it is for 15 seconds. So statement 3 is also correct and statement 4 is also correct. So B is correct. Only statement 2 is not correct. All are correct. Is this question clear till now? So what happens? I provided some of the examples like category. If we talk about milk, so in milk, previously we discussed about the Mycobacterium tuberculosis that its count should go down. But now mostly we take C. burneti also in the count that the target micro. This is a target bacteria which needs to be reduced in the milk products. Then we can say that the if its count is very like is low or in the like it's in the not in the public health risk amount, so we can see that the product is milk is pasteurized. So these are the processing conditions which are being used, like maybe 63 degree or 60 for 30 minutes, like that. Second category of the food is seafood. So there botulinum is basically it is being taken as a target bacteria and sometimes non-proteolytic types B and F. So all these basically what happens when in the food industry, these kind of food products, when they process like milk or seafood, so they see that the they will treat and they will see the F value, D value, Z value, that if these value will be uh, in the limit or they will see that if that Z value is like they consider 8.6. So all these counts will be considered based on the Z values and F values and D values. So in egg products, so this kind certain like treatment conditions are being taken like around 50 to 60 Celsius, and juices like they will be low acid and the high acidic food juices. So salmonella and all botulinum, these kind of the bacteria, target bacteria, they have like seen usually in industry. So in industry, low count reduction, like these terms are being used mainly for pasteurization or for any treatment like sterilization or usually for pasteurization and sometimes in UHT and aseptic processing also. So these are just examples. So you can relate better in terms of industries also. So can we move to next question? This question, match the following one. Any doubts so far? So match the thermal process which is in column 1. So column 1 you are having many thermal processes like blanching, sterilization and pasteurization. In column 2, and so their purpose are given like reducing enzyme activity and complete destruction of all the viable organism and then killing part of the microorganism and human pathogens. So this is very easy question. We already discussed some parts. We discussed about pasteurization and blanching already. Just sterilization we didn't discuss so far. So A, C, A. Okay. Let's see the correct answer. A is correct answer. C is not the correct answer. Let's understand why C is not correct answer. So blanching basically is for enzyme activity. We know that blanching is done for enzyme activity, right? Then we know that pasteurization is part of microorganism and eliminates human pathogens. we already discussed in the previous question. And then sterilization is basically aimed to complete destruction of all the viable organism. So basically what happens in sterilization, there is a complete destruction or elimination of all the viable microorganism, or the like it destroy yeasts, mold, vegetative bacteria, spores also, spore formers also, and it allows the product to store and distribute at ambient condition with extended shelf life. So it in sterilization, all kind of the bacteria and the yeast and the molds and the spores also, they will be killed. So it is achieved by applying high intensity heat. So around 121 degree Celsius, apply for 15 minutes. We know that that is the sterilization standard sterilization treatment to achieve the to in the food product. So there are two type of bacteria, like organism which are of the public health significance, and another is the spoilage causing the bacteria, spoilage causing bacteria. So these are the toxin producing bacteria mainly. So they will be in low acid food with a pH of greater than 4.6, like Clostridium botulinum, and mesophilic. The spoilage causing bacteria will be mesophilic spore formers will be there like this. So this in the sterilization process, it kills all the bacteria and the spoilage causing bacteria or the spores, everyone. So second related to this. So A is correct, not C. So is this question clear? Now let's move on the UHT processing of the milk. For UHT processing, not for milk in general. So which of the statement is correct about UHT processing? So UHT processing basically uses temperature below 100 degrees Celsius. B is it is not suitable for fruit juices. And then C is saying it sterilizes food by heating it above 135 degrees Celsius for a few seconds. And D is UHT processed food, they require refrigeration. Which statement is correct? So Hari Babu is saying C and Vishakha is saying A. Anyone else? A and C. Suraj is also saying C. So we have two C and one A. Let's see. C is the correct answer. Let's understand why not A. So UHT processing, it uses temperature around 135 degrees Celsius for few seconds, like 1 second or 2 seconds, like that. So it is not correct. A statement is not correct, right? So ultra high temperature processing of milk, if we talk about milk, it heated around 135 to 154 degrees Celsius and around 1 to 8 seconds. So temperature will be like very, very high and time of treatment is very, very low. And it is in B, it is saying it is not suitable for juices, whether it is like suitable for juices. So, but in sometimes what happen in citrus juices where limonin are is the compound. So sometimes while processing, limonin the juice be little um, bitter kind of taste, and sometimes like in milk, in UHT milk, which has a shelf life of like 6 months or so at ambient conditions, like so they have burnt kind of flavor. If you have anytime tried, so you must have experienced some burnt flavor in the UHT milk because the temperature is so high that the uh, basic proteins, carbohydrates, they will have some kind of modification and can give the burnt kind of flavor. So C is not suitable is correct, is incorrect, sorry. Then in UHT processed food, it sterilizes food, making it the cell stable. Yes, this is correct that it makes the food product for storing at ambient conditions, as we see in the UHT process Tetra Pak packaging milk products. So next is UHT processed foods do not require refrigeration until open. So until it is not open, it will be good. But once the packet will be opened, we need refrigeration condition. Sorry. Yes. So once it is opened, we need refrigeration condition. Otherwise, it will be fine at room temperature. So statement fourth, that it requires refrigeration, is not correct, and C is correct, that it process around 5 degrees Celsius for few seconds, which is correct. So what happens, as we discussed, that in UHT processed food, deterioration of color, flavor, and texture can be there. We can observe these kind of phenomena like cooked taste in UHT milk, sometimes loss of chlorophyll, then texture loss in the canned food or dehydrated vegetables, and sometimes there will be toughening of the muscles in frozen fish and seafood. Temperature is so high. So in aseptic commercial sterilization, what happens, 130 to 170 Celsius temperature condition is generally referred to as UHT process. So when we are considering the commercial sterilization condition in the industry, so 130 to 170 degrees Celsius temperature we consider as UHT process. It can be used for fruit juices, honey, wine, then creams, soy milk, yogurt, soup, stews. So all these products we can use UHT process. So this table has different examples, like these are the 4 fruits, and then mango nectar is also there. So UHT treatment is applied, and uh, the bacterial count is reduced to certain, like 4.9 log reduction or 2.2 log reduction of yeast and mold. So we can see that when watermelon is being uh, treated with the UHT process condition, so like 135 days for 2 seconds. So what happens? The total flora count, it reduces to reduces more than 3 log reduction, right? Like that. So these are just examples so that you can understand the application part. So is this question clear to everyone? Okay, we have some doubt. Payan, yes, please. Question.
Ma'am, yeah, ma'am, so is the nutrient value is also go down in this process?
Yeah. Yes. Yes.
And the
So what?
taste and
Yeah.
nutrient
So
value.
yes, so what happens? We discussed that in certain compounds, like the flavoring compounds are very heat sensitive compounds. So the compounds which are heat sensitive, uh, they have some kind of loss. So in UHT milk, burnt flavor is there, right? So
Right.
chlorophyll loss is also there in some products. So these kind of compounds, they will degrade uh, because the temperature is so high, but time is very low. Like in few seconds, we are cooling down to normal temperature or below 4 degree. So what happens? There is not much loss, but there will be some kind of color, flavor, texture changes. But in some kind of food products, but not all, like not all in all the products, because the and also the nutrition loss is somewhat, but not very high, because the time we are taking in just seconds.
Okay, so for the fruit juices, what is the best process for preservation?
So it depends on the basically the shelf life, how much we want. So this specific technique, UHT process, if we are doing, the milk is going around 6 months. So you can understand juice process, the Tetra Pak which we do for all these treatments. In all these fruit products, like their shelf life, real juice and all, they have shelf life around 6 months or so. So juices, they will go undergo usually that kind of treatment, like UHT condition. In
Okay, and
industry, this is used. Yeah.
yeah, apart from the shelf life, if we are concerned about the nutrition's value, then which
Mm.
process is more better for the juices,
So
fruits?
UHT is only the better process, since
Okay.
uh, uh, because shelf life is around like 6 months, like that. If we go for pasteurization, sometimes we go for pasteurization of the juices also, but that is for low shelf life. So basically, it depends on shelf life to shelf life. Obviously, if we apply low temperature at low time, that is the best process, because um, they have the lower transition of the nutrition value, or lower loss of the nutrition value, and other properties, sensory properties also. So better is the, that's why to nowadays novel processing are being taken into the consideration, like HPP treated juices, we see in the market, that high pressure processing treatment is basically, it is already in the like industrial level. So all these treatments are novel technologies, are basically preferred than the conventional one. If we talk about the like, and because nowadays before consumers, they were not very much concerned about the like the freshness, that now the consumers want that we need juices which are very similar to fresh, or the just taken from the raw material, like that
Hmm.
fresh juices. But previously the shelf life was very important, but now industries are also, since consumer demand is more for the fresh like juices, so they are going for novel technologies, high pressure treatment, like those.
Okay, so the
Sometimes
novel
the
technique.
cold pressing, yeah, cold pressing are also being used, like high pressure cold pressing. So just juice will be extracted using the high pressure treatment, and then put it into bottle, and high pressure processing only apply to kill the microorganism. No heat movements in the whole supply, production chain even.
So the high pressure cold press system does not um, decrease the nutrients of it.
No, no, not at all. Because high pressure is just, we are giving high pressure, like certain amount, maybe 6,000 megapascal, and they will just kill the microorganisms and not much alter the like very minimal damage will be there in the food product. Like because there, and in that also microorganisms, like they need around 600 megapascal or like that, but the food content like proteins and all, they will degrade even at the higher pressure. So they will not like the protein structure will be intact, and other nutrition quality intact, but the bacteria will go, like they will destroy. So pressure condition they will make in such in high pressure. So where there is no heat, the process keep the food products, I think, in a better way, or more fresh way.
Okay, thank you.
Thank you for asking. Anyone else? Okay. Kyle, can you unraise your hand? If the if I answered your question. So let's move to the next one. Next question about aseptic processing. So following statement is correct about aseptic processing. So first statement is temperature lower than 100 Celsius. B is the product before being placed in a sterile container. C is it does not involve sterilization. D is it is same as canning. So anyone help us or can share the his or her opinion, please feel free to or can unmute and give answer. Both is okay. Both are okay. So B, we got 2 as B. Okay, let's see. B is the correct answer. Product is heated before being placed in a sterile container. So basically, in aseptic processing, the product is subjected to a thermal process. First product, it will be heated. So to inactivate the vegetative cells or the spore prior to being placed in a sterile condition, and the container it will also be sterile separately. So the food product is being sterile separately, then the container is being sterile separately. The food product is being placed into the sterile container in a sterile environment also. So environment will also be sterile. The product and the container or the package are sterilized separately and put them together in a sterilized environment using a form fill seal machine. So it will be like a continuous process where the food product is being heated separately and the container is being sterilized separately, and then finally they'll be put together in a sterilized condition so that no microorganism can grow or can come into the food product or stick to the container. So B is correct answer. This is done to create a safe, ready to eat, shelf stable food product which can be transported even at the ambient condition or the room condition also. Like that, usually the Tetra Pak milk product or juice product, they come in such kind of they will be treated like that. So their milk will be first pasteurized or sterilized, or milk will be treated under UHT processing, and then uh, the container will also the Tetra Pak, they it will also be sterilized using sometimes H2O2, hydrogen peroxide spraying or something like that, and then the milk will be put into the Tetra Paks in the sterilized environment. Then since very high temperature is being applied, so around 130 degrees Celsius to 180 degrees Celsius in aseptic commercial sterilization, these processes are generally referred to as ultra high temperature process. So as we already discussed, that UHT process, which is having 130 to 180 degrees Celsius. So because it will happen in few seconds, so product will be treated in UHT kind of treatment condition and sterilized. Otherwise, the product should be sterilized in 121 degrees Celsius, that to be 15 minutes, which will lose the quality of the food product very high. So better to use UHT process, right? So it uses the temperature lower than 100 degrees Celsius, which is incorrect. And then it does not involve sterilization, very incorrect. So it uses sterilization and it is same as canning. Let's see if it's same as canning or not. So this UHT process are based on enzyme inactivation. Since some enzyme exhibit greater heat temperature range than do microorganisms. So sometimes what happens, enzymes which are not like the which are not good, undesirable enzymes, they will be having higher temperature than the microorganism even. So sometimes target temperature, so the target temperature will be chosen based on the most resistant either the enzyme or the microorganism. So we will see, like we will take the highest cap of the temperature, which is the like either based on the most resistant one. So there will be many microorganisms or the enzymes which will have low temperature, they will degrade, but we will take the highest or the most resistant one in all the treatment, in all the temperature treatments, they will be used. So fourth point is saying that same as canning. So let's understand what is canning, so we can understand why it is not correct. That what is the difference between aseptic processing and the canning. So this is the diagram on the left side bottom. So this is of aseptic processing, that the mostly the fluid or the liquid foods are being taken because the pump, they can be easily pumped like juices or the milk products. So they will go in thermal processing like heating, holding, and then cooling, and then package will be there. It will go sterilized, and here the food sterilized package will be there, and the filling will, and finally closing, and finally after closing the product will be delivered, which can be stable at room temperature even. Now let's see what is canning. So canning is basically food preservation in which food is processed, and what happens, sealed in airtight containers and heated to a temperature sufficient to destroy microorganisms and enzymes that cause spoilage, by achieving commercial sterility and extending life. So in this also the uh, product is sterilized. So what happened first? The product is filled some kind of jars or tin cans, and then uh, air will be removed, and the will be hermetically sealed or tightly sealed. Um, uh, only like then it will be heated like in such kind of pressure vessels. The product will be with the tin, they will be placed in the retorts or the pressure vessels. So there the product will be sterilized with the jar. So in conventional canning, the food is being placed inside the container, is then removed to produce vacuum, and then the container will be sealed, and finally the is can into some kind of retort or sterilization with the help of steam or hot water. This is canning. So this is very different from aseptic processing, right? Like here we are not filling in the aseptic environment. We are first itself, before sterilization, we are filling into the cans or the tins, which is very much different than aseptic processing. B is also incorrect. So B is only the correct one. This is the flow chart of canning. Yes, Vishakha?
Ma'am, as you said that what is aseptic packaging is sterilizing the food, right? And what is
Yes.
canning? The cans are sterilized and we add the food in it. So what's the difference? Main difference?
Both uses the sterilization, but the main difference is, see, in this the flow will be different. That the aseptic, in aseptic condition, food we take and we will sterilize it. Then we will take package differently, separately, and we will sterilize that also. Then we will fill in an aseptic environment. Whereas in canning, we will take some jar or tin first, and we will first itself we will put the food in that food product in that. We are not sterilized food product or jar so far. Then
Some basic
both
as
both will be
sorry, interrupt.
sterilized then at the same time. Basic is same, that both is the sterilization.
Sorry, ma'am. As per my knowledge, while canning, the bottles or the cans which we use, they are also sterilized, right?
The cans which will be first, see, first we will take cans, we will put the food products in that, right? Then we will going to uh, sterilized in the high pressure vessels which are retorts like that, right? Yeah,
Okay.
sometimes yeah, this cans are also being sterilized like that.
Yeah,
Like cans
that's
will,
what.
yeah, you are correct that cans will also be sterilized. And then but we will fill the product in the can which are sterilized, but we have
Okay,
not
that's the basic.
sterilized the food product. Yeah, then in like
Yeah, we have got.
yeah, okay, so we
Thank
will
you,
take,
ma'am.
yeah, okay, sorry, I had a little confusion that uh, can will be sterilized or not. So thank you that you
Fine,
asked.
ma'am. Thank you.
Okay,
Thank you, ma'am.
so we will, okay, thank you. So, if what we will do, we will have the cans. So cans will be first sterilized, as Vishaka said, and washing and sterilization will be done, and the cans will be like the cans will be in this condition where the above one, the from the above it will be open, and from bottom it will be closed, and then these kind of cans in this some kind of syrups or the brine solution will be filled, like which will help to increase the shelf life of the product and lower the water activity. And our food product, it will be like some kind of vegetable if it's there, so it will be like washed and then peeled, or the like some minimal preparation will be there, and that will be blanched, and then that will be filled, and from one side the can will be there. In the can syrup will be filled, and some vegetables or the fruits will be there. After that the vacuum will be released out, so exhausting is the process. Then can will be sealed and then processed, and then finally processed means it will be processed at a high temperature and then cooled, and finally it can deliver as a product. So this is the, so it in this also the cans will be sterilized. Okay,
So, ma'am,
so let's see
basically,
this.
yeah,
Yeah,
basically
yeah,
the in this canning, the food is not processed.
just
Food is
blanched,
not.
minimal process.
Yeah, food is not processed actually. The can is sterilized and it is
Processed.
processed.
Yes,
Okay,
yes,
and
yes,
in the aesthetic, sorry, what it is? Yeah, in the previous one, we sterilize the food also, and we sterilize the can also. We
yes,
sterilize everything, the atmosphere
mm,
and everything. Okay.
yes, yes. In
Okay.
basically aseptic processing, na Tetra packs kind of packing will be there. Most
Yeah.
of the time I've seen.
So in the canning, there will not be the loss of nutrients.
In canning, there is obviously loss of nutrients.
There is loss of
Loss of
nutrient, or
nutrients.
there is loss of
No,
nutrients, but it's not
because
processed now.
because what happens when you are treating with at high temperature in canning, then there will be obviously loss of some kind of nutrients, but not that much. But the shelf life is very high. Like in canned food, you see shelf life is 6 months, maybe 1 year.
Okay. There is still the confusion in both.
What is the confusion?
The confusion is that only you said na, ki the food is not processed in canning, right? It's not heated,
Indeed.
it's not nothing is done. Then how
It
the
is
nutrient value
blanched.
decreased?
See, though in canning, two times product is heated. First in blanching, it is treated, and then when finally product is uh, in the can, it will go in the retort, and this processing is there, right? So what happens in processing? Sterilization is happening.
Mm-hmm. Okay.
So two-time heat treatment is there. So one blanching only, if you do blanching, blanching has lot of nutrition loss, because what we do, we put some kind of vegetable into the hot water, and in hot water, with the hot water, uh, vitamin C and other nutrients, they will lose at the blanching step itself.
Okay. Okay,
So
got it.
in this two time we are using heat. In
Okay.
canning also there is loss of nutrients, but because we have the brine solution and the syrups, which means high, either high sugar will be there or high salt will be there, so that will help to increase the shelf life.
Okay.
Okay.
Thank you.
Okay, thank you. Let's move to this question, next one, about aseptic processing only. Which one of the following statement is correct regarding aseptic processing? So aseptic processing involves both packing and the product, and then it extends shelf life of the product without refrigeration. Third is it is commonly used for juices and dairy products. Fourth is aseptic processing requires high temperature and for longer durations. So we already discussed this aseptic packaging processing. So I think it's very easy for everyone. One, two, and three. B. Let's see. B is correct. Vishakha is correct. So aseptic basically processing, it involves sterilization of both products and the packaging, and then it extends the shelf life of the product without refrigeration, obviously. And we uses juices and dairy products for high flowability and pumpability, and it uses high temperature for short duration, not high duration for long duration. No. So let's now discuss this match the following question about D value, Z value, F value, and lethal rate. So these are the different process concepts we have in column 1. And in column 2, we have the descriptions, like which one is the increase in temperature that reduces D value by a factor of 10. Second is time required for 20% reduction in microbial population. Third is total time required for uh, to accomplish stated reduction. So the total time which is required to reach a certain level of reduction in the population of the vegetative cells or spores at constant temperature. And fourth one is the ratio of F value at reference temperature to F value at any high temperature, at any temperature. So which one is correct match? What is D value, Z value, F value, and rate?
Option A.
Option A. We got two, B and A. Let's see which one is correct. So A is correct, since like we know that D value, which is also known as decimal reduction time, it is the time required to destroy 90% of the organism. So D value is basically the number of the minutes. So D value is the time value. In minute it will come. Required for the survivor curve to traverse one log cycle. So this is the survivor curve. In this 1 log cycle reduction of the microorganism from this to this, we will take this much, how much time it will require as a d value. So it mathematically it is equal to the reciprocal of the slope of the survivor curve and is measured of the death rate of the organism. So d value is equation, if we see, 2.303 by k log x by x naught. So log this x naught is the initial microbial count and x will be after the treatment. So x here will be 0.1, which is 10 log cycle reduction of, so it will be 10 and it will be log 0.1 x naught by x naught. So it will be log 0.1 and d value will be 2.303 by k. This is the d value. So d value from here we can see is time required for 90% of the reduction in the microbial population. So next is the Z value, which is thermal resistance constant. So this is basically temperature value. Z value is a temperature value. So it is the slope of TDT curve. TDT curve is where D value is on the y axis and temperature is on the x axis. So this is the slope of thermal death time TDT curve, and it tells about the number of the degree Celsius which are required to bring about 10-fold change in D value, decimal reduction time value. So it is the temperature which will tell what is the 10-fold reduction in or 1 log cycle change in the D value. Z value is T2 minus T1 upon D log D1 minus log D2. So this is a slope of TDT curve. This. So this will be, here is the T2. Here is the T1. So here is the D2 and here is the D1. So we need to calculate the slope, which is T2 minus T1 upon log, because this is in log scale, y axis. So log D1 minus log D2. So Z value will be the increase in temperature that reduces D value by a factor of 10. So F value is thermal death time. So F value is a time value. So the total time required to accomplish a state reduction in the, so stated reduction. So it says we decided that this much reduction we want, and that stated reduction in the population of the spores or the vegetative cells. When we achieve that stated reduction, we say at that time we reached F value. So this time can be expressed as a multiple of D value. So it's the multiple of D value as long as the survivor curve follows a first order model. For example, if we say that 99.99% reduction in the microbial population, we are stating that this kind of reduction in the population we want. So it will be 4 log cycle reduction, which means F equals to 4D. A typical thermal death time in the thermal processing of the self-stable food F equals to 12D. We take for sterilized food which are we say commercially sterilized, we do 12D reduction, like which usually kills Clostridium botulinum. So we take this value. So F value is total time required to accomplish the stated reduction in a population of vegetative cells or spores at constant temperature. Now is the lethal rate. What is lethal rate? Proportion of the thermal death time at a temperature T to the thermal death time at a reference temperature Tr. So this is like lethal rate curve and time. So we say it is the lethality. This much is the lethality. So under uh, the lethal rate curve is a plot of lethal rate, and when we take the area of this curve, it is known as this much of the lethality is done, or the integrated impact of time and temperature on the microbial population, expressed as time and temperature. So at reference temperature, time is taken, this much is time and this much is the lethal rate, and we take that area under the curve is its lethality. So lethal rate is ratio of F value at reference temperature to the F value at any temperature. Question clear? Can we move to the low temperature treatments like chilling and freezing? Okay, let's move to this question. Which of the following statement is correct regarding the benefits of chilling? Chilling does not extend the shelf life of fresh and processed fruits and vegetables. Chilling is performed at between -18 degrees Celsius to -23 degrees Celsius. Sensory and nutritional properties are minimally affected by chilling. Fourth, chilling involves the removal of both sensible heat and heat generated by respiratory activity. So we have the low temperature or heat removal process like the chilling and freezing, right? So first we are discussing chilling, then we will discuss about freezing. What is the benefit of chilling? D, 3 and 4. 3 and 4 is correct. So what happens? Chilling basically it extends the shelf life of fresh and processed fruits and vegetables. So first one is incorrect. Second is sensory and nutritional properties are minimally affected by chilling. Is correct. Chilling is performed at temperature between 1 to 8 degrees Celsius, which is this is second one. Basically, chilling involves removal of both sensible heat, which is the like when we harvest the food products, the fruits or vegetables, there will be some heat, sensible heat will be there. So chilling basically removes that heat, and also it removes the heat generated by the respiratory activities. So this is also fourth statement is also correct. So D is correct. So the respiratory heat at 20 degrees Celsius and atmospheric pressure will be that the glucose they will be it will take oxygen and convert into the CO2, H2O, and some kind of some heat will be generated, right? So this heat is the respiratory heat, and in chilling, when we are doing chilling, this respiratory heat will be taken. So we have different food commodities like apple, asparagus, cabbage, grapes, and orange. And when they are being stored at different temperature, so what is the heat heat will be there. So at low temperature, the heat evolved will be low, and at high temperature, the heat evolved will be more. Is this question clear? Can we move to this question? The cold sensitive temperature of subtropical fruits is dash degree, while that of tropical fruit is less than dash degree. So there is one question. By using ice, we can rapidly reduce temperature. As we know that ice is widely available, chilling is the most economically one to preserve the fruits. We can do by ice where the refrigerators are not there, but from the ice there will be like water will also come out. So usually uh, after harvesting the in the refrigerator, if you are putting, that is also the kind of chilling treatment, right? So please answer this question. Cold sensitive temperature of subtropical fruits and tropical fruits. What are tropical and subtropical fruits? Anyone? So let's discuss this solution. The cold sensitive temperature for subtropical fruit is 5 to 8 degrees Celsius, and that for the tropical it may be 12 degrees Celsius. So what are the tropicals and subtropicals basically? Tropical fruits are like mango, banana, pineapple, papaya, coconut, like these, mangosteen, jackfruits, like that, sapota. All these will be tropical fruits. So basically, they grow near equator, and they can like they cannot tolerate much lower temperature. So temperature should be always greater than 10 degrees Celsius, even in the coldest month. It should, even in winters, the temperature should not go beyond 10 degrees Celsius. And ideally, the temperature should be maintained at 20 degrees Celsius. So they need warm, humid climates, uh, and they don't need much frost or low temperature. So these are the tropical ones. And in tropical ones like the citrus fruits, grapes, figs, pomegranates, lychee, guava, all these are subtropical fruits. So they grow in the region near the tropicals, but uh, they are the like they can tolerate mild winters, like they can tolerate 10 degrees Celsius temperatures and hot summers also, but they cannot withstand severe frost. So it's just in which climate they are growing. They are tropical, subtropical, like these kind of fruits are being divided. So there will be some temperate fruits also, which can grow in temperate condition. Now let's talk about chilling injury. So which statement is correct about chilling injury? Chilling injury occurs at temperature above freezing point, and it is easy to detect during cold storage. Injury only affects tropical fruits, and chilling injury affect membrane function of plants. Any answers? A. So yes, you guys are correct. Chilling injury occurs at temperature which is above freezing point. So chilling injury is basically a physiological defect in the plants when there and because of that there is reduce of the quality and the product loss of the product utilization, because when it is fruit is being exposed to low temperature but greater than freezing point or non-freezing temperatures like 5 degrees Celsius, but not the freezing point temperature above the freezing point, it can happen. This kind of injury is chilling injury. So first option is correct. The cold sensitive temperature of the subtropical fruits is 5 to 8 degrees Celsius, while that of tropical fruit less than 12 degrees Celsius. It is given that in subtropical fruit, 5 to 8 degrees Celsius temperature, it is the like a cold sensitive temperature, and for tropical, it's 12 degrees Celsius. So the chilli, it affects fruits and vegetables, both in which are grown in tropical, subtropical regions. C is incorrect. Then it is not easy to detect during cold storage. So when the product is stored in the cold storage, chilling injury cannot be detected. But when the product comes at the room temperature, the symptom be appeared. So B is incorrect. Then chilling injury, basically it induces lipid membrane lipid phase transitions, uh, which lead to loss of the membrane and the physiological dysfunction. So it may damage membrane functions of the plant cells. So D is also incorrect. It can also result from the direct effect of the reduced temperature on enzymes or the indirect effect of the membrane perturbations on intrinsic enzymes. So because of chilling injury, there can be effect in the enzyme, and because of enzyme, there will be effect in the membrane and the texture of the food product also. So overall loss of the food product can be there. It is also speculated that lipid peroxidation can also occur, and that can injure the food product when the low temperature stress will be there. So like this kind of symptoms we can see in the food product, like there will be softening, browning, pitting, or uneven ripening when it is undergone chilling injury. So highly sensitive food products, they include banana and pineapple. And so if we talk about in depth about the chilling, there can be two biochemical mechanism in the chilling injury. So what happens? One is the like lipid transition. So in liquid crystalline, in the lipids are in the liquid crystalline state. So in the chilling, when there is chilling stress, so the lipids in the cell membrane, they will convert from lipid crystalline to solid gel-like structure, and because of that, there is loss of the integrity of the membrane. And because the solid gel-like structure will form, what happens? There will be high permeability of the cell membrane, and more electrolyte leakage will be there, and because of that, cell damage or cell lysis will happen. Also, because of solid gel formation in the cell membrane, more Ca plus 2 cytosolic Ca2 plus will form, and free acids will form, and finally the acids will go under peroxidation, and the cell membrane will also oxidize, and cell damage will be there. And third mechanism is that because of the solid gel formation in the cell membrane. Functional disturbance of the cell membrane proteins will be there, and there will be low photosynthesis and respiration, and the reactive oxygen species will form, and because of the reactive oxygen species are there, so fatty acids will go under peroxidation, and or cell membrane can directly undergo oxidation, and finally there will be lysis of the cell. These are the like diagram. So in banana we can see that browning is there because of chilling injury. In avocado we can see pitting is there, so in the circle area, and in the cucumber we can see these watermarks, so water spots will be there. This kind of we can see when the fruit is undergone chilling injury. Now let's discuss this question. Which of the following statement is correct regarding freezing? Freezing involves reduction in temperature of a food above its freezing point. Second, immobilization of water as ice lowers the water activity. Third, microbial deterioration is eliminated during freezing. And fourth is freezing increases the rate of enzymatic and chemical reactions. Anyone? So Suraj is saying B is correct answer 1, 2, and 3 about freezing. So Suraj, C is correct answer 2 and 3, not 1. We know that freezing involves reducing temperature of food below its freezing point. So here it's above freezing point, so which is incorrect. Then water in the food, it becomes first crystal, and the immobilization of water as ice and the resulting concentration of dissolved solutes in unfrozen water lowers the water activity, and the temperature is usually minus 18 degree or below. So this is incorrect, and the immobilization of water as ice lowers the water activity, which is correct, and microbial deterioration is eliminated during freezing, obviously, because minus 18 microorganism will not able to grow and can regular cell activity. And then freezing, it increases the rate of enzymatic and chemical reactions. So it decreases actually. So it is incorrect. So microbial deterioration is low because at minus 18 microorganism will not able to grow. Then reduce the rate of enzymatic and chemical reactions, and higher shelf life, convenient to use, and efficient handling and transportation. Because the frozen product is in the solid form, so easy to handle also. So we know that this is the curve of heat removal on x-axis and temperature on y-axis. So when heat is removed in the water, so this is the water one. So first sensible heat will be removed, and then product will go under supercooling state. Means product is in liquid form, but um, it is at very low temperature. Then all the latent heat will remove and the ice crystal will form, and finally the sensible heat will remove further for storage. This is for water, but in food product there is little bit different than the uh, water, pure water, because food has many components like proteins, carbohydrates, fats. So food curve is like that follow the same trend but a little bit low. So everyone is okay with so far discussion? Can we start drying now? So let's discuss drying. Which of the following statement is correct about drying? Drying of moisture to reduce water activity. Drying always requires high temperatures. Drying increases the weight of the product, and drying do not affect the shelf life of the product. So answer A is correct, that drying is the means that there is removal of the moisture to reduce the water activity. So it is basically a unit operation where liquid, solid or semi-solid feed will convert into solid product, and its water activity will be low and moisture content will also be very low. Drying is basically the removal of moisture to reduce water activity, which is correct A statement. It can be done at various temperature, not always at high. So this is high. Then drying increases the weight of the product, but it reduces the weight of the product. And drying basically it increases the shelf life by reducing the microbial growth and enzyme activity. So this is also incorrect. B option. So A is correct answer. So what happens basically if a food product is there, like this is a food product. So if we supply heat, so heat transfer is there, and because of the heat, water transfer will be there as a water vapor out of the product. So this is mass transfer also. So both heat transfer and mass transfer is taking place in drying process. Okay. So let's discuss one numerical problem. The weight of water in a sample is 50 grams and the weight of the bone dry matter is 200 grams. Calculate the weight percent wet basis percent moisture content of the sample. Please try to solve this question and let me know the answer. Let's see the solution. So what is happening here? It is given in the question that the weight of water in a sample is 50 grams and the bone dry, which is completely dried product, is 200 grams. And we need to calculate wet basis percentage moisture content. So moisture content in wet basis is mass of the water divided by mass of the dry sample or total mass into 100. So mass of the water is given as 50 grams. So 50 divided by mass of the water, which is 50, plus mass of the dried product, which is 200. So 200. So which is 50 divided by 250 into 100. Which, because we want into percentage, so into 100. This is 5. 100 divided by 5 is 20%. So B is correct answer. Is this question clear? Can we move to this next question about freeze drying? So which of the following statement is incorrect regarding freeze drying? Freeze drying involves sublimation of ice under reduced pressure. It results in the removal of both bound and unbound water. C is uh, freeze dried products have a high shelf life, and D is it is less effective than traditional drying methods. So we get fourth. Vishakha is saying fourth. Okay, let's see. Only four. She's correct. So what happens in the freeze drying, which is also known as lyophilization, is basically a drying process which involves crystallization of the solvent or suspension medium at low temperature and then sublimation. So what happens here? First, product will be stored at -80 or -30, so ice will be there, and then suddenly the sublimation is there. So no liquid form, directly gaseous form will be there. Because we are doing this at low pressure. Then sublimation is directly into the vapor phase, and final moisture content will be less than 10%. So this method is used to produce self-stable products that remains unchanged after reconstitution with water. So after reconstitution, we have seen that the freeze-dried fruits are there. So when we and many other things freeze-dried forms are there, like some kinds of soups. Some kinds of soups are also there. So when we put water in that, it gets original shape. So all the ice, water, free water, and some bound water must be removed. In this, there are 3 steps. So what happens? The product will be there, which is in the ice form. So the freezing of the product is there under atmospheric condition first. So all product will be in ice form. Then primary drying will be there. So what happens? Ice sublimation will be there at reduced pressure. So at low pressure, pressure will be low, and ice will be sublimation. Ice sublimation will be there, and secondary drying after the ice sublimation will be there. Then what happens? Secondary drying process will happen where the bound water will be removed by the desorption drying. Drying of the product will be there to get the final humidity. So we have this product, and there are different zones in this form, like sublimation front. Sublimation front is formed, which is also known as transition zone. It is basically the interface between frozen zone and the dried zone. So when the product is drying, so what happens? In some product, some part moisture already removed, which is dried zone, and another part will be the frozen material, frozen ice material. So the interface is known as the sublimation front. In the heat transfer through frozen layer, so from frozen layer, heat transfer is there. Then heat and mass transfer both take place from inside to outside. So what happens? Heat transfer from conduction, it happens like hot to cold region. This heat of diffusion process. So the rate of heat transfer depends on basically the thickness and the thermal conductivity of the ice layer. And this basically this is based on the Fourier's law. So what happens? The rate of the heat transfer basically is directly proportional to the thermal conductivity of ice cross sectional area, which is Q equals to minus Ka T by dx. T is temperature change, which is the difference of T1 and T2. K is the thermal conductivity of the material. A is the area cross-sectional, and dx is the distance, how much it is traveling. So heat rate of heat transfer is directly proportional to the thermal conductivity of ice, cross-sectional area through heat is being transferred, and the temperature difference between the heating surface and frozen layer, but inversely proportional to the thickness of frozen layer, which is dx. So this is the how the heat is transferring in the freeze drying process. So in this, only fourth is correct, which is that it is less effective. Sorry, fourth is incorrect, which is less effective than traditional drying. Others are correct that it is produce high shelf life product. It removes both unbound and bound water, and it also uses the sublimation of ice under low pressure. Is this question clear? Can we move to spray drying process?
Ma'am, in the both,
Yes.
yeah, in the both drying and freeze drying, both the new, what about the nutrition's value?
Drying, which kind of drying you are saying?
The heat
Drying and
drying?
freeze drying. Heat
Yeah.
drying, sun drying or some, okay.
Yeah, sun drying or electric drying.
Uh, so in freeze drying, the high amount of nutrition will be there, and the product is in the intact form, more intact form, because there is like ice. First product is cooled, like ice is formed, and then directly at low, because we are applying here low pressure. So at low pressure, what happens? The boiling point of the water, it will be at low. So at low heat, all the water will convert into sublimation state.
Okay, so the nutrition's value will stay like the same.
Will be high in like it will be same in freeze drying, and when we do sun drying or other kind of drying, there some kind of heat sensitive compounds like vitamin C can degrade.
Okay, so the drying is not also a appropriate process.
No, freeze drying is very good process.
Which one?
Freeze
Freeze
drying,
drying.
yes, yes, it's
Okay.
where
Can you
that's
can you
why
give me, can you give one example of freeze drying, please? The
uh,
process, any one material and how it get freeze drying.
sometimes in the market shelf, we get the slice of the apple and many different kind of products like fruit slices and like that. They are in freeze dried. There it is being written on the packet: freeze dried apple slices or freeze dried citrus slices, like that. Many times, if you have seen on the in
Okay, so
marketplace.
okay, so that slices how they do the freeze drying. One example only. If you explain one example, I can understand the what is exactly freeze drying.
So what happened is, like any product is there, like we are talking about apple slice, okay? So apple
Hmm.
slice will be there. We will put it into -80 or
Okay.
-30, anything, okay? All the
Hmm.
water in this, all the water in the cell, it will freeze, it will convert into ice crystals, okay?
Okay.
Then, like usually, what we do is we put into -80-degree refrigerator this apple slice for maybe overnight, 12 hours, okay?
Hmm.
Next day, we will keep in the freeze dryer equipment. So what happens? There will be uh, one slab will be there. On that, you will put your like apple or something.
Hmm.
Then what happens here? In this, pressure is low, vacuum is maintained. Okay, so your
Hmm.
this boiling point of water will shift towards lesser side. Okay, it will not be 100. It will be maybe 20 or 30 or 40, but it will not
Hmm.
be 100.
Hmm. Hmm.
Okay, so what happens? The crystals, the ice crystals, they will directly go in vapor phase, water vapor. So this
Okay.
ice will go. Now what is left? You have apple.
Right.
You have this inside texture like this. Maybe here void will be there. No water. Void only void will be there because water is already gone out.
Okay. So nutrient value also remains the same.
Yeah, mostly like that's why the this is the most recommended method, and in cell studies like microbial studies also freeze drying is being done when there is RNA DNA level study.
Okay, and after this process, can we keep in the refrigerator or we have to, can we keep as the normal temperature?
Normal temperature we can just put into one package like pouch, and we can
Okay.
just seal that pouch and sell.
Okay, so at the normal temperature.
Yeah, because water is already gone. So it is a dried
Okay.
product. So dried product has no water, so no microbial growth.
Okay, and is this the process done by the nitrogen gas or this that is different?
So what happens if you take nitrogen in cryo cooling? If you listen, liquid nitrogen.
Hmm.
If you take liquid nitrogen, what happens? It liquid nitrogen is having very low boiling point. So what happens eh, when you like usually when you dip like if you're dipping this apple slice into liquid nitrogen, this all water will be converted into ice within maybe within 2 to 3 minutes. All it will be a frozen product. You need not to put into -80.
Okay.
So liquid nitrogen is for conversion into any product. It will convert it into ice like brittle.
Okay, and there is no change in the nutrients value if you use the nitrogen gas.
Yes, yes.
Okay, okay, thank you.
Nitrogen gas, it is like when you take even your product out or something, then what happens, nitrogen gas is like continuously foam will come out. If you open nitrogen if you put nitrogen gas in your maybe container, some maybe glass or something, what will happen, it will continuously the foams will come out. The liquid will convert into the gaseous form because it has very low boiling point. So it is as soon as it comes into ambient condition, it starts boiling even.
Okay,
So the product, it will not leave any residue on the product also.
okay, thank you.
Okay, let's move to this next question about spray drying. So which of the following statement is correct about spray drying? It is only used for solid foods. It involves drying the product in a vacuum chamber. It is suitable for heat sensitive products, and it uses a stream of hot air to dry the atomized liquid. So what is correct about spray drying? Spray drying is basically is used for liquid or the solid, semi-solid food products. So there what happens, like this kind of setup will be there. So here atomizer will be there and flowy material will be there. It will go into the spray drying chamber. So for the because of the atomization, so atomization means that what is happening, like here is gone. The product is coming and going through these nozzles kind of thing. So liquid jets will be there, and it will come atomize, and then what happen, convert it into the dry powder by rapidly drying with hot air stream. So from in this chamber, hot air, this air heater, so from this hot air is also coming. So stream of liquid is coming like this, hot air is going like this. There will be this hot air will take all the moisture, and the product will be dried and come at the bottom. Like this is the process of spray drying. Okay, so it involves basically a vacuum chamber. Sorry, it does not involve a vacuum chamber because normally air is supplying, so no vacuum is required. And then it is not suitable for heat sensitive products. If the product is heat sensitive, so spray drying is not preferred. So D option is correct, that it uses a stream of hot air to dry the atomized liquid. And C is not correct, that because it is not suitable for heat sensitive products, and it involves drying the vacuum chamber, which is incorrect, and not solid products, liquid products. Okay, so usually we see that skim milk powder is produced from this method. Whey, casein powder, cheese powders, ice cream mixes, mixed powders, egg powders, cereal powders, dried infant foods, instant coffee, tea powder, juice powder, soup powder, fish powder. Like these products, they will be made in spray drying method. So what is happening here? Atomization is happening of the liquid feed, and then that will go into the drying chamber. In drying chamber, there is like air. So there is a contact of the air stream and hot air stream and the food product, and because of that, moisture will be removed from the food product, and dried product will come at the bottom, which will be separated, in which will go in the cyclone separator, and product will be like separated into the cyclone separator based on the density. So limitation is that product which is having high sugar and organic acids, they are difficult to dry because they'll go and stick in these walls of the spray dryer, and they'll make cake agglomerates and the cakes, and heating temperature is greater than 100 degrees, so loss in the thermolyable compounds like vitamin B1, C, anthocyanins, or phenolic compounds like lycopene or probiotic bacteria. So they'll not use for heat sensitive compounds. Next is the drum drying. So which statement is correct regarding drum drying? Drum drying is you taking the rotating device to dry product. It is suitable for high viscosity product. Results in the formation of fine powder, or the product is applied as a thin film on the drum surface. Please let me know. So let's see. Vishakha is saying D, 3 and 4. Anyone else about drum drying? So what happens in drum drying is this kind of technology. Answer B is correct. 1, 2, and 3 are correct. So drum drying is a contact drying method. So what is happening? Here, high viscosity products will be suitable, and there will be this drum which is of metal, and it is heating because there is condensed steam at the inside, and the product will be on outside, which is being dried. So the wet product is applied as a thin film on the drum surface, and the dried product is removed from the drum. So either from here, dried product will be taken out using a blade. So here, sheets like or the flake like products will get, not fine powders. The cylinder is heated by steam condensing inside, so which is at a pressure of 200 to 500 kilopascal, and the temperature of the cylinder wall is around 120 to 155 degrees Celsius. So like this, product is scraped from blade. So drum using the rotating device, which is correct. High viscosity product correct. Product thin layer is applied as a thin layer on the drum surface, which is correct, and it results on the formation of fine powder, which is incorrect because we get flakes or the sheets, not the powder. Let's match the following. Here are the sun drying, spray drying, drum drying, and freeze drying in the column 1, and in column 2, key features are given. So this is very simple now. Please let me know. So Vishaka is saying B, Suraj is saying B. Okay, both B. B is correct answer. That the sun drying uses natural light for drying. Spray drying involves atomization of the liquid into the hot air stream. And then drum drying, it takes rotating drum for drying the product. And freeze drying, it involves sublimation under the reduced pressure into a hot air stream. Okay, so this is B option. Let's see the last question for today. Which of the following statement is correct regarding quality changes occurred in the food product during thermal processing? So unsaturated lipids are more prone to oxidation than the saturated one under heating in the presence of oxygen. Second is level of total and available carbohydrates are largely unaffected during thermal processing of fruits and vegetables. Then third is minerals are susceptible to change in bioavailability due to interaction with other food components. Please let me know what is your answer. 1 and 3. C. No, D is correct answer here. That 1, 2 and 3 all are correct. So what is happening is when so when we are heating the food product, so there is the lipids. They will basically the unsaturated lipids. They will be more prone to oxidation than the saturated one under the presence of heat and oxygen. So this is discussed already by Mishra sir in the lecture videos. This kind of table you can see. Then carbohydrates. So the level of the total and available carbohydrates are largely unaffected during the thermal processing of fruits and vegetables. So carbohydrates will be unaffected. Lip Unsaturated lipids will go oxidation, and minerals, let's see. So minerals, they are susceptible to changes in the bioavailability due to interaction with other food components. So all three are correct. So D is correct answer. So we discussed mainly today about the basic principles of the processing and the preservation, and we discussed about the factors affecting microbial growth like pH and water activity. And then we discussed about the processes which add heat, like we discussed about the blanching, pasteurization, then sterilization, aseptic processing, UHT, canning, and then we discussed about the thermal process calculation, D value, Z value, F value, and lethal rate. And then we discussed about heat induced changes in the product quality. We discussed also about the heat removal methods like chilling, freezing, and chilling injury is the problem in chilling. And we discussed about removal of water, like we discussed about drying, and we discussed about the moisture calculation, like wet basis, dry basis, Dry basis is also like you can probably get, you can solve some questions on dry basis. Then we discuss about freeze drying, drum drying, sun drying, and spray drying methods. In next lecture, we will be discussing about cleaning and washing, sorting and grading, peeling, coring and slicing, containers and packaging materials for fresh produce, and packaging method and equipments. So yes, so we were talking about one in the during the lecture that this lecture slides will be available on this Google Drive link. Okay, and the recording of today's class and last two classes you will also get in my YouTube channel. And if you want to ask some question, please join Q&A group in the course. And if you have some questions now, you can stay, or if you don't have any question, you can leave.
Ma'am, is this links will available just right now here? We have to copy it or what?
So in the recorded lecture, you can see that these links will be available.
Okay.
Okay. And if you want I should paste all these links in the chat box. I can do that for you guys. Do you want me to paste these links in the
Yes.
chat box? Okay,
Yes.
I'll paste.
Yes, ma'am.
So all the lectures, like on the same day, by today itself, I'll be uploading these PPTs and the recorded video lecture by today itself. Okay, so you can, whenever you're free, you can go, you can see all these as soon as possible for your revision. Where is the chat box? Okay. So I've pasted all the both of the link here, both these link here in the chat box, so you can access. And also in the recorded video, it will be there. Also in the this PPT, in the PPT also it will be there in the Google Drive. So you can access anytime. Okay, so let's meet on next Sunday, which is on 17th. So same time. If you have any other doubt, you can stay, or thank you, or you can leave if you don't have doubt. Please join the next session, and thank you so much for asking the questions and joining, taking your time out.
Okay. What will be the next time? What will be the next time?
Same 4 to 6.
6. Okay,
Yeah,
actually,
my
ma'am,
all
it was my
lecture.
first lecture.
What happened?
Actually, it was my first lecture, and I haven't attended anything else before. That's why I have lots of questions. I'm sorry for that.
Oh. Oh, it's good that you have asked so many questions. Like, it's good. I want people to interact, and because it's not a like one side discussion class. It's like an interactive session. So if you have any doubts, you should ask. That's why these lectures are being held. Otherwise, there's no need to have these kind of lectures even.
And in the chat box now, where there is three dots and where the chat is there, I when I click on it, it says something is wrong.